Hi viewers, in this class we are going to discuss cytokines. In general, the development of an effective immune response involves lymphoid cells, inflammatory cells and hemopoietic cells. The complex interactions among all these cells, that is lymphoid and inflammatory along with the hemopoietic cells, they are mediated by a group of proteins collectively designated as cytokines. Cyto means cell, and kinin means to move. It is derived from the Greek language. Generally, all these cytokines secreted by WBC or other cells. Except the RBC, many other cells can capable to produce the cytokines. Usually, the secretions of lymphocytes are called as the lymphokines, and secretions of monocytes and macrophages, they are usually known as the monokines. Whereas the interleukins are the cytokines that are secreted by some leukocytes and act on other leukocytes. These interleukins designated as IL. Till now, many interleukins from IL-1 to IL-29 and more than that have been described. Some of them known by their common names like the TNF, tumor necrosis factor. Whereas chemokines are group of low molecular weight cytokines that affect chemotaxis and other aspects of the leukocyte behavior. These molecules play an important role in inflammatory responses. And moreover, we should know the terms lymphokines and monokines. Lymphokines and monokines not limited to the lymphocytes and monocytes and extends to broad spectrum of cells and types by which they get produced. Coming to the introductory part of the cytokines, the macrophage migration inhibitory factor, MIF, was identified in 1966 by John David and Barry Bloom. Later on in 1969, Dudley C. Dumande proposed the terms lymphokine and the monokine. These two terms were proposed by Dudley. And later on in 1974, Stanley Cohen proposed the term cytokine. Till now we are using it. Later on, Ogawa described the importance of early acting growth factors, intermediate growth factors and late acting growth factors. Coming to the properties of cytokines. What are the properties attributed to cytokines? Generally, these are the low molecular weight molecules which have low molecular weight. Cytokines have the low molecular weight. If you observe the chemical nature of the cytokines, they are either the proteins or the glycoproteins. Chemically, they are proteins or glycoproteins, and most of them they involve in regulatory functions. And if you observe the mechanism, cytokines bind to specific receptors. On the membrane of target cells on which actually it is going to act, and thereby triggering the signal transduction pathways that ultimately alter the gene expression in the target cell. The susceptibility of the target cell to a particular cytokine is determined by the presence of specific membrane receptor on the target cell. And if you observe the dissociation constant for cytokines and their optimum receptors range from 10 to the power of 10 to 10 to the power of 12. And they can mediate the biological effects. Cytokines can mediate the biological effects at the picomolar concentrations. Picomolar concentration of cytokine is enough to mediate the biological effect. And cytokines regulate the intensity and duration of the immune response. They regulate the intensity and the duration of the immune response by stimulating or inhibiting the activation, proliferation, and as well as the differentiation of various cells, and by regulating the secretions of antibodies and other cytokines. Then they also influence numerous immune cells. Cytokines influence numerous immune cells. That is, the cytokines secreted by even a small number of lymphocytes activated by antigen can influence the activity of numerous cells involved in the immune response. Let us see best example for this. The cytokines of the T helper cells. Cytokines produced by activated T helper cells can influence the activity of the B cells, that is B lymphocytes, TC cells, cytotoxic T lymphocytes, NK cells, natural killer cells, HSC, hemopoietic stem cells, macrophages, and even the granulocytes. So cytokines of activated T lymphocytes, TH cells, they influence all these cells which are usually involving in the immune responses. See here, this is the cytokine producing cell. Upon a stimulus, the stimulus may be, in general, it is a antigen. If there is any stimulus, it acts upon the cytokine producing cell, and thereby these cells produce cytokines. These cytokines act upon, bind to the receptor of the target cell, and upon binding they mediate signal transduction and finally alter the gene expression of the target cell that elicits a particular biological effect. So this is the cytokine and the general mechanism of action. And cytokines exhibit different kinds of activities. This is related to activity of cytokines, namely autocrine activity, paracrine activity, and endocrine activity. What is the autocrine activities? Auto means self, crine means secretion. Auto means self, crine means secretion. So the secretions of a particular cell, otherwise cytokines which are produced from a particular cell, and these cytokines act upon the same cell again, auto, self. And in the paracrine, the cytokines which are produced from a cell, they interact with the neighboring cells. They interact with the neighboring cells. And whereas in the endocrine mode of activity, the cytokines which are produced from a particular cell, they released into the bloodstream, they travel along the way of the circulation and interact with a target cell which is far away from the cytokine producing cells. So autocrine means self-reacting, paracrine act upon the neighboring cells, whereas the endocrine act upon the long distance cells. This is about the activity of the cytokines and attributes of the cytokines. There are major 5 attributes, namely pleiotropic action, redundancy, synergism, antagonism, and the cascade induction. Pleiotropism, otherwise pleiotropic action, is a given cytokine that show different biological effects on different target cells. The redundancy, 2 or more cytokines that mediate similar functions. Redundancy makes it difficult to ascribe a particular activity to a single cytokine. Whereas synergism, the combined effect of 2 cytokines on cellular activity is greater than the additive effects of the individual cytokines. Antagonism, the effects of one cytokine inhibit or offset the effects of the another cytokine. And finally, the cascade induction, the action of one cytokine on a target cell induces that cell to produce one or more other cytokines, which in turn may induce other target cells to produce other cytokines. There is a serial activation or serial induction takes place. See here the pleiotropism or pleiotropic activity, that is phenomena of different actions produced by same cytokine on different targets. Here there is a activated T cell, Th cell, and it produced IL-4 interleukin 4, and it acts upon the B cell for the activation, proliferation, differentiation. It can also act on the thymocyte for the proliferation. It acts also on the mast cell for the proliferation. This is the pleiotropic activity of the cytokines. Whereas redundancy, different cytokines have the same effects on same targets. Here activated Th cell, T helper cells can produce IL-2, IL-4 and IL-5, and the target cell is the B cell, and all of them show the repeated action of the proliferation. The third one is the synergism. It occurs when the combined effect of 2 cytokines on cellular activity is greater than the additive effects of individual cytokines. That is, additive effect is greater than the individual effect. Activated T helper cells produce interleukin 4 and interleukin 5. So the combined effect is greater than the individual effect. Thereby, they induces the class switch to IgE, production of the IgE type of immunoglobulins. And antagonism, it is the opposite effect, inhibitory effect. The effect of one cytokine inhibits the effect of the other cytokine. Here activated T helper cell produce IL-4, interleukin 4, and thereby the activity of the interleukin 4 is inhibited by interferon gamma. So thereby blocks the class switch to IgE induced by IL-4, interleukin 4. So here it is a antagonistic effect, inhibitory effect. So one cytokine inhibits the effect of the other cytokine. And finally the cascade effect. Cascade effect is nothing but cytokines can stimulate the production of other cytokines in a serial manner. Here activated T helper cells are there, produce interferon gamma. It acts upon the macrophage and in turn to produce interleukin 12. It acts upon the activated T helper cells, and they are thereby the activated T helper cells produce interferon gamma, tumor necrosis factor, interleukin 2 and other cytokines. So cascade effect is a serial induction method. One by one thereby they activate and produce some other factor to activate another cell. So these are the attributes of the cytokines. See here there is a total diagrammatic representation of all these attributes: pleiotropy, redundancy, synergy, antagonism, and the cascade induction. And if you observe the similarities, differences among the growth factors, hormones, and the cytokines, cytokines share many properties with hormones and growth factors. The distinction between these three classes is often blurred. All three are secreted soluble factors that elicit their biological effects at picomolar concentrations by binding to receptors on the target cells. These three mediators differ in their modes of expression. Growth factors tend to be produced constitutively, whereas cytokines and hormones are secreted in response to discrete stimuli, and secretion is short-lived, generally ranging from a few hours to few days. Whereas hormones act at long-range distances in an endocrine fashion, cytokines act over short distances in an autocrine or paracrine fashion. Hormones produced by special glands and show unique action on few types of target cells. Cytokines are often produced by and bind to variety of cells. So these are the similarities and differences among the growth factors, hormones, and the cytokines. And furthermore, the activity of cytokines was first recognized in the mid-1960s in the supernatants derived from in vitro cultures of lymphocytes that contain soluble factors that could regulate proliferation, differentiation, and maturation of the immune cells. It was discovered that production of these factors by cultured lymphocytes was induced by activation with antigen, and biochemical isolation and purification is generally hampered because of their low concentration and the absence of the well-defined assay methods. Development of gene cloning and discovery of cell lines provide the simple assay systems, and furthermore, monoclonal antibodies have made rapid quantitative immunoassays for each of the cytokine. Then coming to the cytokine families, they belong to four families, namely hematopoietic family, interferon family, chemokine family, and TNF tumor necrosis factor family. There are more than 200 types of cytokines were discovered. So that is the proteins with cytokine activity, they are included under these four families. And also you can observe the cytokine receptor families, to which actually all these four families of cytokines come and contact to the receptors of the target cells. These receptor families are basically 5. There are 5 cytokine receptor families are there. There are 5 cytokine receptor families, namely immunoglobulin superfamily receptors, class 1 cytokine receptor family, it is also known as hematopoietin receptor family. Third one is class 2 cytokine receptor family, it is also known as interferon receptor family. And fourth one, TNF receptor family, and fifth one, chemokine receptor family. So don't get confused, these are the families of cytokine receptor, whereas these are families of cytokines itself. So we should not get confused. See here the cytokine receptor family images. This is the immunoglobulin type receptor, and this is the TNF receptor family. This is a cytokine class 1 or type 1. This is cytokine class 2 or type 2, and this is the chemokine receptor family. So these all are the membrane bound receptors which are ready to attach with certain cytokines. So this is a cytokine, and here the cytokine binds. And functions of the cytokines. Generally, the cytokines are responsible for the hematopoiesis by producing the CSF, colony stimulating factor, and they mediate inflammatory reactions. The interleukin 1 and tumor necrosis factor plays a major role in the inflammatory reactions, and they tend to show chemotaxis, that is interleukin 8, macrophage inflammatory protein 1 MIP1, and BLC, B lymphocyte chemoattractant. All these factors, IL8, MIP1 and BLC, all these they exhibit chemotaxis or chemotactic mechanism. And immunostimulation due to the presence of IL-12 and interferon gamma, they show the immunostimulation. Coming to the next one, immunosuppression. IL-10, interleukin 10 is responsible for the immunosuppression activity, which is tend to use during the organ transplantation. Angiogenesis, here there is another factor, VGGF, vascular endothelial growth factor, that is useful in the angiogenesis process. And embryogenesis is mediated by TGF-B and lymphotaxin. So these are the main functions shown by the cytokines. And general functions: development of cellular and humoral immune response, induction of the inflammatory responses, regulation of hematopoiesis, control of cellular proliferation and differentiation, healing of wounds. Although the immune response to a specific antigen includes the production of cytokines, cytokines act in an AG non-specific manner, antigenic non-specific manner. So they are not at all specific for a particular antigen. So they show similar mechanism against all the kinds of antigens. Then coming to the medical use of cytokines as drugs. Bone morphogenetic protein, BMP, it is used to treat bone related conditions. Erythropoietin, EPO, used in treatment of the anemia. GCF, the colony stimulating factor, used to treat neutropenia in cancer patients. GM-CSF, neutropenia and fungal infection in cancer patients. Interferon alpha, used to treat hepatitis C and multiple sclerosis. Interferon beta, to treat multiple sclerosis. Interferon gamma, to treat chronic granulomatous disease and osteopetrosis. And interleukin 2, IL-2, to treat the cancer, and IL-11, interleukin 11, to treat the thrombocytopenia in cancer patients. So these are the different kinds of cytokines which are usually used as drugs for the treatment of various disorders in the medical field. So this is the list of different kinds of cytokines and interleukins and their serial number, source of the interleukins, function in the malignancy, cancer, and different kinds of cellular pathways adapted by all these interleukins. So some sort of reference also given in a tabular form. And coming to the cytokine storm, it is a recent phenomena. what is the cytokine storm is? Cytokine storm is an umbrella term encompassing several disorders of immune dysregulation characterized by constitutional symptoms like systemic inflammation, multi-organ dysfunction that can lead to multi-organ failure if it is inadequately treated. In general, a healthy cell produce, a normal cell produce, it is a healthy cell and it is an infected cell. An infected cell produce interacted with the cytokines produced by the active immune cell. An active immune cell has the capacity to produce cytokines which interact with the infected cells rather than the healthy cells. Now, whenever there are more active immune cells, the secreted cytokines of all these immune cells, they are not only interact with the infected cells but also interact with the healthy cells and cause damage to that of the healthy cells. Then the patients with cytokine storm, are febrile, fever may be high grade, fatigue, anorexia, headache, rash, diarrhea, arthralgia, myalgia, neuropsychiatric findings. These symptoms may be due to cytokine induced tissue damage, acute phase physiological changes, immune cell mediated responses, some of the times that leads to ARDS. ARDS is the acute respiratory distress syndrome. Acute respiratory distress syndrome. See here, due to the cytokine storm that is stimulated by pre-existing inflammatory conditions and the COVID-19 infection nowadays. So that leads to more production of the cytokines that results in the clotting, shock, lung injury, cardiac damage, immune paralysis, renal failure. So due to the immune failure, organ failure and the secondary infection, usually death occurs in the individual. In severe cases of cytokine storm, that is renal failure, acute liver injury, cardiomyopathy can also develop. The combination of renal dysfunction, endothelial cell death and acute phase hypoalbuminemia can lead to capillary leak syndrome, and finally death occurs in the individual. So it is very dangerous one. That is a cytokine storm. It may damage lungs, alimentary canal, brain, heart and cardiovascular system, liver, kidneys and circulation and even the eyes. So due to the multi-organ failure, renal failure and usually death occurs in the individual. This is all about the cytokine storm. There are different kinds of cytokines which are used as therapeutic techniques in therapies. They are in clinical use like Enbrel, Remicade or Humira, Rofiran, Avonex, Actimune, Neupogen, Epogen. So they are used in different kinds of diseases like rheumatoid arthritis, Crohn's disease, hepatitis B, Kaposi sarcoma, feline leukemia, multiple sclerosis, CGD, osteopetrosis, increase in neutrophils, reduce infection, cancer and AIDS patients, increase RBC production. All these are treated with different kinds of cytokines. They are used in clinics. So this is all about the cytokines and their families, receptor families, and the uses of the cytokines. Thank you one and all.